Hello, everyone, and welcome to today's webinar, Visualizing Cancer Biology Through Cell Culture Inserts. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Rayees Ahamed. Rayees is working as an R&D scientist having 6+ years of experience in cell and protein biology. He is focused on developing protocols and workflows for various cell biology applications. For a complete biography of today's speaker, click their name in the presenter window at the top right of your screen. Rayees, welcome. You may now begin your presentation.
Hello everyone, thanks for joining today. My name is Rayees Ahamed. I'm an R&D scientist at Thermo Fisher Scientific. In today's webinar, I'll be talking about the applications of cell culture inserts, specifically focusing on in the area of cancer biology. So I'm going to discuss more in detail about how we can effectively use the cell culture insert system to model multiple cell types. Thereby we can study different events of cancer. So before we dive into the cell culture inserts, let's recap some basics of cancer cells that defines the characteristics compared to normal cells. Every cancer cell starts with a mutation in the genetic code of the cell, which corrupts the entire cell. So upon proliferation, these corrupted cells divide and increase in number that enlarges the tissues around. This localized state is called hyperplasia. Followed by it leads to the abnormal growth of tissues and organs around, and at this point it is called dysplasia. So later these cells are benign to their local environment, that's why we call them tumor. But the real problem starts when these corrupted cells tries to escape their original environment, thereby invading other parts to the body. When it becomes malignant, it adds more complexity for our understanding of these cancer cells due to the properties such as migration of cells from one place to another, invasion through different physical barriers, and then they tend to induce angiogenesis, such as forming their own blood vessels to get continuous supply of nutrients. So why cancer cells are so complex? Over the years of research, many scientists have been figured out different principles of cancer cells, trying to understand various events of cancer. Among all, the hallmarks of cancer described by Hanahan and Weinberg were really helpful and acted as a blueprint for researchers to dissipate the cancer cells mechanism. More importantly, these hallmarks helped us to model these in vitro as well as so that we can understand them, experiment them, and intervene different therapies to stop these hallmarks at an early stage of cancer. So out of these hallmarks, the most challenging hallmarks which are highlighted here that are difficult to model in vitro are studying cancer cell migration, invasion, and then tumor-induced angiogenesis. So how do we study these cancer phenomena in the lab? Conventionally, we have well-established culture systems such as monolayer, where we can grow single type of cells just to grow them and then do simple assays such as proliferation assays and drug treatments. But we cannot study events like cell-to-cell interactions, and it's basically lacking the actual tumor microenvironment. So thereby, it's very difficult to study two different cell types in a monolayer culture. And then we have co-culture models where we can culture two types of cells together, which helps somewhat to understand their interactions. But at times, these cells are difficult to separate if you want to analyze them individually. Also, it may require two different media if you are culturing two different cells, so which is a limitation for this model. And then we have third 3D culture models, which are very emerging, such as spheroids and organoid based cultures, which mimics our in vivo like environment. Yet again, we do have limitations as it is still emerging, and they are difficult to reproduce and interpret their results. Also, these may not be very feasible to study hallmarks like cell migration and invasion. So we need an alternative culture system which is very well established. So how do we solve this? So the idea here is, instead of just co-culturing two types of cells in a monolayer, so we can make use of co-culture with interfaces, which allows us to grow two different types of cells in a single vessel with a physical barrier in between them. So these are named as interface cultures. So what you see on the right hand side are regular cell culture well with an insert at the center. So these inserts are called as cell culture inserts, which basically divides the culture well into two different compartments, an apical side and then basal side, with an porous membrane in between. So this porous membrane helps in movement of cells, nutrients and growth factors between the two compartments, just like our capillary system. So this is an excellent tool to mimic tumor environment, especially to study events like cell-to-cell interactions and migration of cells. Since each cell type is physically separated from each other in this system, unlike the regular culturing, it's not cultured together as monolayer. So typically these interface cultures are classified into two types. The first one is liquid-liquid interface culture. That is the most widely used in many applications. And the second one is called air-liquid interface culture, that are primarily used in modeling the respiratory cells of lung. So how are these cell culture insert system helping to study cancer phenomena? So if you see on the left hand side, the progression of tumor, which involves events such as migration and invasion in response to various growth factors, and then it also leads to tumor induced angiogenesis and then paracrine signaling between different cell types. So all these hallmarks are easy to model with the use of cell culture inserts, and I will demonstrate how do we model these with an examples in upcoming slides. So in real world cell culture inserts, how do they look? Well, what you see here is called Nonsell culture inserts in a carrier plate system developed by Thermo Fisher Scientific. So it is a two compartment multi-plate system consisting of cell culture inserts with a porous membrane, a carrier plate and a regular cell culture plate at the bottom. So the membrane inside the cell culture inserts comes in three different pore sizes to suit different experiments. Also, it is height adjustable, so the carrier plate height can be adjusted to increase or decrease the distance between the carrier plate and the bottom well. So this provides flexibility in handling the volume of media required in each application. So this is the disassembled view of lung cell culture inserts in a carrier plate system. So as you see in the picture, it has the regular multi-well plate as the lower chamber, carrier plate to hold inserts as a upper chamber, and we have removable cell culture inserts which can be placed at the center of the carrier plate. So at first, I would like to demo how to perform migration of cancer cells in response to different chemoattractants or growth factors. So this is a quick step guide to perform a migration or invasion experiment in response to chemoattractants. So just to give a difference between migration and invasion. So when we say migration, so the cells tend to migrate just in response to the chemoattractants, Whereas in invasion, they have to physically cross an ECM barrier in the culture plate, thereby they invade and pass through the other side of the plate. That's why we call them as invasion. So looking into the steps, first we need to select the right type of insert and pore size, and then the hanging height of the plate should be adjusted according to our media requirement. Second, we have to feed the lower well with media containing the chemoattractant or growth factor of our choice. Then we have to seed the cell suspension on top of the insert. Then we have to incubate the cells as do we regularly incubate our monolayer culture. So what happens inside the insert after incubation? So due to the presence of the chemoattractant, the cells tries to cross the insert barrier by migrating from top of the insert to the bottom of the insert, as seen in this graphical representation. So this is an example of breast cancer cells migration in response to growth serum at the bottom of the plate. So simply we have cultured MCF7 or MDMB 231 cells on the top of the insert, and serum containing media were placed at the bottom well, and cells were actively migrated from top to bottom, and the migrated cells are stained with crystal violet, and the data has been depicted here. So as you can see here, the migrated cells are stained in blue in the wells which contains the serum, and the wells without serum did not help in migration. So next we have demonstrated another application in which the migration of tumor associated macrophages in presence of colorectal cancer cells has been demonstrated. So we have cultured the tumor associated macrophages on top of the insert and colorectal cancer cells at the bottom. Thereby we assayed different applications such as their wound healing capability and invasion capacity, and then one of the important cancer hallmark, that is the epithelial to mesenchymal transition, which is called as EMT induction. First to generate the tumor associated macrophages, so we have used the human leukemia monocytes called THP1 cells, which are treated with PMA. Upon they differentiate into tumor associated macrophages as seen in the images. So morphologically you can identify the difference by looking at them from monocytes to macrophages. So this tumor associated macrophages are seated on the inserts and the colorectal cancer cells are seated at the bottom well. So after they reach certain confluency, so we made a scratch to create a wound, and the influence of tumor associated macrophages in the wound healing has been assayed here. So what you see here is the assay performed on HCT116 cell line, so which is an colorectal cancer cell line seated at the bottom of the well, and then tumor associated macrophages seated on top. So the results shown to help in the results show here the tumor associated macrophages shown to help in complete the wound closure, whereas when there is no presence of tumor associated macrophages, the wound is not healed. The same experiment has been done with another colorectal cancer cell line, which is called HD29 cells. The similar results has been observed in wound healing. As you see, in presence of tumor associated macrophages, there is a complete closure of wound in the HD29 cells. So next, to assess their invasion capacity, so what we have done, so we have created an ECM barrier by coating the inserts with an Gelrex. And then we seeded colorectal cancer cells on top of the insert and tumor associated macrophages at the bottom. Thereby the colon cancer cells could able to invade the ECM barrier at the insert, and they can migrate to the bottom of the cell culture inserts, Upon migration, so these cells has been stained with crystal violet, and the results what you see here shows the difference in the cell population when they were cultured together. So as I mentioned, the epithelial to mesenchymal transition is an important marker in understanding the migratory and invasive properties of cancer cell. Hence, we assess the gene expression changes once we culture these tumor-associated macrophages together with colorectal cancer cells, which shown to demonstrate key changes in some genes when the colon cancer cells are cultured together with macrophages. Okay, so the next important application we have demonstrated with cell culture inserts is modeling the endothelial cell migration and in vitro tumor-induced angiogenesis. So at first what you see here to model the endothelial cell migration, so we have used HUX cells seeded at the top of the insert, and in the bottom well we have seeded well-known angiogenic factors such as BFGF and VEGF containing media, and when these conditions are established, HUX cells in response to BFGF and VEGF, they are actively migrated from top of the inserts to the bottom of the insert. So after migrations, these cells are stained with crystal violet, and as you can see in the result, there is a strong migration of HUX cells seen in presence of these growth factors compared to their control. So next, to create an tumor angiogenesis model in which we can establish a culture with two different cell types. So basically the tumor cells influence of endothelial cells to form tubules can be modeled with this cell culture inserts. For that, what we have done is we have used an ovarian cancer cell type which is called SKOV3. They are proven to release VEGF in their microenvironment, and this VEGF is recognized by HUX cells to form endothelial tubes. So as a quick step guide, so the steps to model this in a cell culture insert system. First, you have to select the right type of insert, and we have to coat the bottom well with Geltrex ECM layer because the HUX cells require the ECM coating to form endothelial tubes. Followed by the HUX cells will be seated on top of the coating, and the next step would be we can see the cancer cells on top of the insert. So now we have established an dual cell culture dual cell type culture system where you have a cancer cell on top of the insert and you have an HUX cells in the bottom well. So upon incubation, what happens, as you can see on the right hand side image, so inside the insert, these cancer cells, which are SKO3 cells, tend to migrate to the bottom of the insert, and then it secretes VEGF in the bottom well that enables the HUX cells to respond to form endothelial tubes. So these are the results of the tumor angiogenesis model we have established in the insert system. As you can see, it has developed beautiful endothelial tubes. The far left image shows in the absence of cancer cells, HUX cells fail to form tubes, whereas in the presence of SKO3 cells, they actively form tubes because of the growth factor VEGF released by the cancer cells, which happen due to the paracrine signaling of the migrated cancer cells at the bottom of the inserts. So one advantage of using this cell culture inserts while imaging with fluorescent dyes, since these two different cell types are in two different layers, you can stain them with two dyes and you can image them in a single plate only. So as you can see here, the green dye here represents the HUX cells, and the red dye represents the cancer cells at the insert. So the last model I would like to demonstrate is a triple co-culture system, that is modeling three different cell types using the cell culture insert system. So here we tried to model the bone metastasis in vitro using the cell culture inserts. Typically in a bone metastasis condition, it involves three different type of cells. The first one is the tumor cells, second is endothelial cells, third the bone cells. So the tumor cells actually invade the ECM barrier at the tissues and interacts them with the endothelial cells, which leads to the transendothelial migration of these cells, thereby metastates the osteocytes at the bone. So we try to mimic this system using our cell culture inserts. So to perform this experiment in the insert system, first we cultured the osteocytes at the bottom bell, and then second we cultured the endothelial cells at the bottom side of the insert. So this is a tricky step where first we need to coat the surface of the insert with an ECM such as Geltrex, and then the insert is kept in an upside bottom position to seed the endothelial cells. And an overnight incubation helps in othering these endothelial cells to the insert, which are grown until they become tightly confluent to create a physical barrier. So at last, we seed the tumor cells on top of the insert, thereby achieving a triple co-culture system. And all these three different type of cells can be stained with different fluorescence dyes, and they can be individually imaged under a fluorescence microscope. So these are the results of the triple co-culture experiment we have done. So we can see the transendothelial migration in presence of tumor cells and endothelial cells and osteocytes, whereas in the absence of endothelial cells, there is no transendothelial migration is seen. So the difference here is captured by staining both the cells in two different fluorescence dyes. The red depicts the endothelial cells and the blue depicts the osteocytes. Okay, so beyond the cancer biology applications, these lung cell culture insert systems can be used for a variety of applications, such as transport studies, mainly to name drug transport across epithelial and endothelial barriers, and to study drug transport across brain microvascular endothelial cells, such as we can even create a blood-brain barrier model using the cell culture inserts. And then it can be also used for tissue engineering, like angiogenesis. We can use them to model different epidermal and epithelial tissue models, which help in skin tissue modeling. And then we can do other regular co-cultivation studies to study any two different type of cell-to-cell interaction. And also we can study cell substrate interaction with this cell culture inserts. So to know more about the detailed protocols of the applications described here, you can visit our cell culture inserts page, either by scanning the code on the right hand side, or you can simply visit thermofisher.com/cell culture inserts. Also, we have a dedicated cell culture learning and resource center where you will get better guidance on choosing the right cell type, media, and reagents tailored to the cell culture inserts. Well, that's all I had for this webinar. Thank you everyone for your kind attention, and I'm happy to answer any questions.
Thank you, Rayees, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. We'll answer as many questions as we have time for. Now let's get started. Our first question here, what factors should be considered when selecting the appropriate cell culture insert for a specific experiment?
Okay, thank you for the question. So typically when you design an experiment, you will be choosing a particular type of cell. So that will be your first factor. So it depends on what type of cell you use. You can choose the kind of dish you want to use. So Generally, these lung cell culture insert system comes with different formats, such as it comes in 24-well format, 6-well format, also in 12-well formats. So depends on the size you require for your experiment. You have to choose first the format of your plate, and then the most important thing will be the pore size of the cell culture inserts, because the pore size varies depends on the type of assay you do. For example, if you are doing an transport studies, such as studying chemoattractant assay from top to bottom, that will require a smaller pore size, for example, zero 0.4 micrometer. But if you are doing migration and invasion like studies, and it depends on the size of your cell, you have to choose your pore size. Suppose if your cells are very big, you can go up to 8 micrometer pore size. So it varies according to your type of experiment you perform. So the second most important thing will be choosing your pore size. And third will be, once you initiate your experiment, so you need to choose how much of volume of media you're going to use in the culture insert. So depends on that. As you seen in the slides, you can choose different heights in the cell culture inserts. So you have low, medium, and then highest position. So if you're keeping it for longer days, you can use the highest position, and then if you're using for a couple of days, you can go for the lowest one. So these are the general factors to consider before you choose cell culture inserts.
Great, thank you. Another question here, do cell culture inserts affect the growth and behavior of cells compared to monolayer?
Technically, no, it won't affect the growth and behavior of the cell. The reason is because these cell culture inserts are also made up of the polycarbonate material, which is the one we regularly use for the tissue culture treated flasks. So both are manufactured with the same material, so it will enable the attachment growth just like in your regular flasks. So it won't change or affect the growth and behavior of your cells.
Thank you. Our next question, can cell culture inserts alone be used with different types of cell culture vessels?
So currently there is no universal design of cell culture inserts available. So brand to brand, company to company, these cell culture inserts differ. So what you've seen here is lung cell culture inserts. So they are compatible with lung cell culture carrier plates. So if you order that carrier plate system, so if you technically see, there will be a multiple plate, and then there will be a carrier plate. On top of that, there will be an insert. So they are specifically manufactured to fit in that carrier plate. So you can't actually use the cell culture inserts with some other brand or some other type. But one tip would be, so all these three components can be ordered separately. Suppose if you have a multiple plate and carrier plate already, you can retain them and just you can order the cell culture inserts alone. Or if you have cell culture inserts, you can order the carrier plates alone. So you don't need to order all three always whenever you start an experiment. So that will save you some cost. But otherwise, they are compatible to the respective designed vessels.
Thank you. Another question here is, how do we image cells with different hanging heights in cell culture inserts? And will it affect the focus and resolution?
Yeah, that's a good question. So that's an important question as well, because while you image at different heights, definitely it will affect your focus and resolution. So for that purpose, what we recommend is, so suppose if you are using a highest position in your cell culture inserts, so before you image, actually you have to bring it down to the lowest position. So in order to reduce the space between the bottom of the plate and your insert. And the lowest position, the gap between the lowest position is less than 1 millimeter. So when you keep your inserts at the lowest position, technically it will not cause any focus or resolution problem. And we have tried with multiple experiments, it did not give any issues. But if you keep them at like in a medium position or highest position, definitely it will not focus it, since the distance between the bottom and the cell culture insert is more. So in that case, we always recommend bring down your inserts to a lowest position, and then you can image it. That will solve the problem.
Thank you. Some great questions. Just a reminder, if you have any more, please go ahead and enter them into the Ask a Question box. Another question we have here is, how do we detach cells from cell culture inserts? And is there any different protocol used?
No, we don't use any different protocol. It's just like how you do in your regular multiple plates. So you can use the common dissociation reagents like trypsin or EDTA to detach them. As I mentioned, these are made up of polycarbonate, and then we don't alter any protocol in their dissociation. So right away you can just add them, and then they will detach after a few minutes, and then you can go ahead and do as you do in your regular cell culture. So we did not prescribe any specific protocol for this, so you can stick to the one what you have already doing in your lab.
Thank you for that. Another question, can we reuse the cell culture inserts after an experiment?
No, they are not designed for multi-use. So these are made for single-use experiments. So generally, any plastic, when you try to reuse, that can seriously contaminate your culture. So, and it is not autoclavable. So these are all made for single use. So you can use one time, and then you have to use fresh one when you perform an experiment next. So these are not recommended to use multiple times. So it will definitely affect your the upcoming experiments you do.
Thank you. Next question, can cell culture inserts be used for 3D cell culture experiments?
Yeah, that's a good one. But right now, we haven't explored anything on 3D cell culture experiments. The reason is, as you know, 3D cell culture experiments typically tend to suspend your cells in the form of spheroids and organoids. So these cell culture inserts are actually cell culture treated just like your regular flasks or multiple plates. So if you see them, like it will go and just attach to the bottom, so it will not give that suspended culture. So we haven't really experimented anything 3D wise in this cell culture inserts. But if you want to try, I can recommend, like, maybe your bottom multi-well plate can be an ultra low attachment plate. So you can just swap the regular multi-well plate. Instead of that, you can use a ultra low attachment plate. That may help to suspend your 3D samples only at the bottom, but definitely you can't suspend them in the cell culture inserts since they tend to attach to the membrane. Currently, we don't have any protocols or recommendation to use these cell culture inserts for 3D cell culture.
Great, thank you for that insight. And thank you so much, Reyes. Do you have any final comments for our audience?
Yeah, if you have any more questions or like queries regarding the cell culture inserts, or if you don't know how to use them for a particular experiment, so we have more protocols and detailed recommendations available in our website, as I shown the URL. So you can go to that, and then you can refer our documentations, and or you can reach to our customer support team. They will help you as to how to use these non-cell culture inserts for your experiment.
Thank you again, Reyes, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. But before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today and those submitting during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you, everyone.